The goal of this study was to understand which brain structures are involved in odor attention. At any given moment, the brain is bombarded with information from the environment, encoding sounds, lights, touch, and smell. And to do anything, we must filter out all of the irrelevant information and focus only on the senses that are important to the task at hand. Previous work has shown that the medial prefrontal cortex is crucial for this filtering process called selective attention, but we do not know how the medial prefrontal cortex communicates with the olfactory system to attend to specific odors. Here, we map the connections between the medial prefrontal cortex and two olfactory structures, the olfactory tubercle, which has been previously implicated in olfactory attention, and the piriform cortex, of which its role in attention is unclear. As shown in Figure 1, I injected rats with two viruses into two prefrontal subregions, the prelimbic cortex and the infralimbic cortex. The viruses encoded either a red or green fluorophore tagged to synaptophysin, a protein that is specifically expressed in synaptic terminals. The prelimbic region was injected with the green-expressing virus, and the infralimbic region was injected with the red-expressing virus, so we know that any green fluorescence indicates the synaptic terminal of a prelimbic neuron, and any red fluorescence indicates the synaptic terminal of an infralimbic neuron. After injection and tissue collection, I imaged the brain regions of interest with a microscope and used the imaging software to automatically quantify the green and red synaptic terminal densities in the tubercle and the piriform. Referring to Figure 2, I first found that the tubercle receives significantly more input from the infralimbic cortex than either the anterior or posterior piriform. After finding this, I wanted to know what specific areas within the tubercle were receiving these inputs. In Figure 3, I compared the synaptic terminal density within the medial and lateral tubercle and found that the prefrontal cortex primarily targets the medial tubercle. Specifically, there were significantly more infralimbic inputs to the anteromedial tubercle than to the anterolateral tubercle, shown in Figure 3, Panel D. I further examined which specific layers were receiving these inputs, and in Figure 4 showed that infralimbic projections are primarily targeting layer 3 of the tubercle. Finally, Figure 5 shows the quantification from mapping the synaptic terminals within the piriform cortex. I found that both prelimbic and infralimbic efferents target anterior over posterior piriform. These inputs were significantly denser in anterior piriform layer 2. Overall, the results of this study suggest that the medial tubercle is the primary region involved in olfactory attention, and I hope to further investigate this finding in the future using physiological and behavioral experiments.